for coming along. The speaker at this CCH seminar is Lauren, our amazing and well-respected research facility coordinator. Let me tell you a few things about Lauren's past. So Lauren completed her honors degree from University of Queensland in 2012 while working as a research technician for the Queensland Centre for Emerging Infectious Diseases. And she worked with the National Hendra Virus Transmission Project. And after that, she was a PC3 molecular diagnostics technician at the Biosecurity Sciences Laboratory. And currently she is doing a part-time PhD here at USQ. This presentation is part of her PhD work, but it's also a very good introduction for all of you in the whole genome sequencing facilities that we have here on campus. So I'm greatly looking forward to know more how to use these facilities. Lauren, the floor is yours.
Thanks, Levy. Good morning, everyone. Thanks for coming along. Yeah, look, I'm really happy to be able to give this presentation today. So, like the title suggests, it's about the minION, and really it's just a beginner's overview to minION. So, for me, this is something that I've been working on for a couple of years, particularly with Oxford Nanocore, minION before that. And I just really felt like this was a presentation that I would have really liked to have seen before I started. So it's taken me some time, particularly in the last year, to get this to work for fungi. And I always felt like it would be great to see something like this before I got started. It probably would have made my life a bit easier. So what I wanted to do today was more give you a beginner's overview of the minION, how the technology works, and also give you some tips and tricks that I've found along the way that have actually made this a bit easier for me so that you don't have to find out the hard way like I did. So I've got a couple of disclaimers first. So first of all, I'm not an Oxford Nanocore expert, but what I can do is give you the information that I've learned over the last few years. So I don't have all the answers, but I'm more than happy to help. If you guys have questions, if you need support in NGS, I'm more than happy to help. And in fact, that's part of my role within the Institute, is to support you through NGS. So actually my job is to manage the facilities, as most of you know, and that includes our beautiful facilities down here in the precinct. It also includes up in Dbook, so we've got our liquid handling robot, which has been a lot of fun lately. We've been doing some work on that. Also the Illumina MySeq, which we've had for about 4 years now. And of course, up in the top corner there, our minION. So these are the facilities that I manage. Not necessarily an expert in sequencing, but I'm happy to help. So that's my first disclaimer. The second one is a really important one for me. This is my 3-year-old Ava. For those of you who haven't met her, she's decided at the moment that sleep is an optional extra. So some of you will understand that. I'm running on very little sleep, so please excuse me if I struggle to get through all of these slides. I'll do my best though. So just a little bit of a background. So in terms of sequencing, what we're familiar with, first, second and third generation sequencing. So historically, I guess the first generation was your Sanger sequencing, and everyone can hear, that's good. And this was largely something that's been done out of house. We haven't had a whole lot of access to. And then came along second generation, which we all probably know as next generation sequencing. And that brought in Illumina, and we're all familiar with Illumina. So for us, we've got the MySeq up there, and that's part of next generation sequencing. Third generation has brought in PacBio and Oxford Nanocore technology, which is, for this presentation, is the minION. So we have both of these here up in Dbook. Illumina MySeq, Oxford Nanocore minION, and they work really nicely together. One's based on short read sequencing, the other is based on long read sequencing. So it's really nice to actually use them together, and you can actually use them for a hybrid assembly. You can use them for assembly to try and polish off your long read. So it's nice to actually have both of these platforms up in Dbook to use together. So a bit of a difference there. So what's the main difference? And there are quite a few, but the main difference, and the one that we want to focus on today for this presentation, is around the fragment size. So when it comes to Illumina MiSeq, it's a short read sequencer. So you're going to get reads that are between 150 and 300 base pairs long. So they're really short. With minION, and this is where it really comes into its own, you're producing really long fragments of DNA. And so what that means is that when you're doing a whole genome assembly, you get lots of overlap with your really long reads. When you're doing MiSeq, you've got these short reads, you don't have much overlap. And so what's going to happen is if you try to assemble a genome, you're going to end up with lots of little gaps because that overlap is just not great enough. minION essentially can resolve that problem by creating long fragments, lots of overlap, and assembling a complete genome. Now, a lot of people say, well, what's the trade-off there? And yes, there is a bit of a trade-off. Essentially you are trading off in accuracy. So when you get these long fragments, often the error rate is a bit higher. And so often what we do now is we use these for a hybrid assembly, like I mentioned before. So you can come back in with MiSeq, do the short read sequencing, and use that higher accuracy to polish off your long reads. And that can obviously increase your accuracy for your long read whole genome assembly. So this talk, like I said, focuses on the minION and the long reads, and that's what we'll focus on, and I'll give you some information about how you can go about getting nice long read fragments for your sequence. Right, so Oxford Nanopore technology, how does it actually work? It is very different to Illumina and Illumina's platforms, and regardless of what platform you use from Oxford Nanopore, they all work the same way. So essentially what's happening here is you have a double strand of DNA, and through a library preparation process, what you do is you ligate on a sequencing adapter to your DNA. And then the motor protein, which is this green globule that you can see sitting in the middle of the screen, it actually captures that sequencing adapter and it transports it down to the nanopore. The nanopore is that blue structure at the bottom, sitting inside an array. So that's within the Flowcell of the minION. So once the motor protein has transported that DNA down to the nanopore, translocation starts, it unwinds and it feeds through the nanopore and down through the array. So through that array, there's actually an electric current that is constantly passed through there. As the DNA passes down through it, it creates this trace. So it disrupts that electrical current, and each DNA base causes a different trace. So A, G, T, and C all produce a different picture, and the software in the background can read that, analyse it, and base call it. So depending on what trace, or what base passes through, there'll be a trace associated with that. The software analyses it and calls what base it is. And then it just assembles, as you can see in that picture, it just starts assembling that base call. So regardless of what platform you use, that's how the technology works. So platforms, as with all sequencing, there's quite a few different platforms, and these are really just associated with ramping up or scaling up how many samples you want to process or what application you're doing. So there's quite a few different platforms in the family. The smallest one is the Flangle, and we actually have access to this now in Dbook, so you can use the Flangle for your sequencing. It's quite small. It's suited to cost-effective and frequent analyses. So this little guy isn't going to generate heaps of data. It's really if you just want to answer a really quick question that only needs a small amount of data to do so. Very cost-effective. These Flowcells only cost $90. So you can answer some questions really quickly and really easily with these. You're only going to generate about 1.8 gig of data, so not much. That's up to. So you're probably not, unless you've got a really good sequencing run, you're probably not even going to get that much. So these are really small. I've actually found these are great for QCing your library before you put it onto a minION. And I'll talk a little bit about that later. So the next one up is the minION, and this is the one that I'll be talking about today. We've got this up in Dbook as well. So this has really been marketed as a portable sequencing device. So it's about, it can fit in your hand, it's really small. You can take it with you into the field. It hooks into your laptop with via USB cable. So it only weighs about 87 grams, so it's quite small. You've got consumable flow cells, powered by USB link. You've got 512 active channels. So that's where the sequencing happens. The next one up is the Gridiron. This one essentially is 5 minIONs combined together. So it works exactly the same as the minION, same flow cell as the minION, but there's 5 of them that can plug into the Gridiron. You can sequence with one flow cell, you can sequence with 3, you can do 5. So you can use them independently or you can run them all at the same time. So at this point, you're really starting to ramp up with your data. You can get up to 150 gig of data with the Gridiron. The flow cell still costs $900. Once you get to the stage, if you are ramping up samples, you can buy them in packs and the cost comes right down. So it does get cheaper. These guys have up to 2,560 active channels. So there's lots of channels there available for sequencing. And the last one is their ProMethine. So this guy is huge. You're going to be generating up to 8 terabytes of data with this. What are you going to do with all that data? I don't know. You'll be there for years, but this guy is really big. The flow cells are different. They're much smaller, but much more expensive, and they've got a lot more active channels. So I think up to 3,000 active channels on a really small flow cell. So there's lots of channels there available for sequencing. Up to 144,000 active channels in total. This guy also comes with its own compute module. So this is going to generate a lot of data. Your flow cells are worth $2,000. If you buy them in packs, you can get them for under $1,000. I went to a workshop a couple of years ago down in Sydney at the Garment Institute, and they had one of these in their laboratory. And I can say that the computer that they had sitting underneath the bench running this thing was about 5 times the size of the actual ProMethine. So they've got data storage all underneath their lab benches, cables, computers. So once you start getting up to this size, you've got to, I think the problem actually becomes what do you do with all that data and how you store it. We're nowhere near generating this kind of thing yet though. So the minION. So this is what we've got up in Dbook. What are the capabilities? There's about 56 different library preparation kits that you can choose from. So things like assembly, species ID, comparative genomics, structural variation, methylation, SMP pooling, and transcriptomics. So really flexible. You can do RNA, you can do DNA, you can do kits that are PCR free, so you remove that PCR bias. If you don't have a lot of DNA, there are PCR kits that you can ramp it up as well. So it's really flexible. A couple of other things, the flow cells for this, unlike MySeq, they're actually reusable. So for whatever reason, if you start a sequencing run, you run it for 6 hours, and you've got enough data there to answer your question, you can actually stop it. So these are designed so that you can just run them for as long as you need to before you can stop it. You don't have to run it for the full 72 hours like you might with MySeq. You can actually stop these, wash them, and store them, and use them again. The other thing that you can do, that is really useful is if you've actually got a really crappy sequencing run, you've started it and gone, oh God, this doesn't look good, something's gone wrong. You can stop it straight away, wash it, and start your library prep again. So in that way, you're not wasting your flow cell. These things are worth $900, so you don't want to just be chucking them in the bin. So yeah, cost effective in terms of being able to reuse them. There's a couple of other kits that are available. So the wash kit that I talked about to allow you to reuse them. You can barcode as well. So if you don't need high amounts of coverage for a minION, you can barcode samples and run multiple samples in one run. And the storage kits, like I talked about, you can rush a buffer back through them, put them in the fridge and leave them there until you're ready to do another run. The other thing that's really cool that's just recent, well, maybe in the last year or two that's come out, you can now refuel a flow cell. So this means that if for some reason you've overloaded a flow cell with DNA, Transporting that DNA down to the nanopore and translocating it through requires energy. And so if you overload a flow cell, you might actually find that you're going to deplete that energy that's available, and that translocation is going to drop right off. And you'll see that in your quality data while you're running it. And if that happens, sequencing is just going to stop because it's run out of energy. What you can actually do now is refuel them mid-sequencing run. So you can just take a bit of this ATP out of the kit, load it into the minION, and you'll see that your translocation speed will come straight back up. So you don't have to lose that on your sequencing run anymore because it's run out of energy. You can just refuel them and it will get back to sequencing, which is really cool. So moving into actually doing some sequencing on the minION. There's a couple of steps that come with it, and this is what I'm just going to go through today. High molecular weight DNA extraction. This is really important, and it's really important to get it right. If you can't get this to work, there's no point in doing anything else afterwards. So you need to get high molecular weight, and you need to get really good quality high molecular weight DNA. The second thing that is what I think is the most important thing is quantification and quality ratio checks. You do need to get quite a bit of DNA to get this to work, a lot more than what you would need for MySeq. And you need to have really high quality pure DNA. Less so with MySeq, it can handle contaminants better. minION doesn't handle contaminants very well. You're going to find that if you don't have pure DNA or RNA, you're going to cause issues on your flow cell and it's going to die really quickly. So you need to make sure that you purify your DNA if you're not hitting your quality ratios. Check your fragment size, that's really important. There's no point in doing this if you don't have long fragments, because that's what we're targeting. If through your extraction you've managed to unfortunately shear up your DNA, you probably don't want to go any further. So check your fragment size, and you can do that. We have a bioanalyzer in Dbook, or you can just run a gel. So make sure you check your fragment size and find out what size your fragments actually are. And then you want to do a high molecular weight DNA purification. So make sure it's a high molecular weight kit. That's really important because a lot of purification kits will shear your DNA. So you need to choose a high molecular weight. And like I said, don't skip this step. If you do have contaminants or impurities, make sure you purify it. If this is all going well, you can then move on to doing a short read eliminator. So like I said, we're targeting long sequences, long fragments. So what we want to do is get rid of all the short fragments. Invariably through those processes, you're going to shear your DNA up a little bit. And so what we want to do at this point is try and get rid of as many of those short reads as possible. It's going to make your analysis at the end much easier. Library preparation, and then flow cell loading, so putting your library onto the flow cell, and then QA check. So once you start your sequencing run, you want to hang around for a little while and make sure that it's actually working. So high molecular weight DNA extraction. I won't hang on this one too much because it's really dependent on your sample as to which extraction you choose, but please choose a well-published high molecular weight extraction method. You want to make sure that you're not going to shear your DNA up through the process, so make sure you get this one right. We've found that freeze-drying samples before you grind and lyse them has actually really improved the DNA concentration outputs. Have a bit of a play around with your sample input as well. I've done that, and I've found that you can actually push those sample inputs a little bit higher and you get more DNA at the end. Essentially what you're trying to do is get as much DNA out of this as possible. You do need quite a bit of DNA to get through all these processes. Don't vortex. Vortexing shears up your DNA. Don't do it. The last stage that I vortex is when I lyse the sample. That's fine, make sure it's lysed properly. After that, don't vortex. Don't pipette mix either. That will shear your DNA. You need to do mixing really gently, and it does add time and it takes a bit of time to get through, but mixing it really gently is going to be really effective. If you can, use wide bore tips. We all know that pipetting shears up your DNA and you can't get away from pipetting through this, so use wide bore tips if you can and be gentle through all of the processes. The other thing that is important is that if you do an ampure bead cleanup as part of your extraction, specifically for purifying, make sure that you choose the correct ratio for long reads. Ampure beads are also used as a size selection, which means you can actually select for short reads. So just make sure that if you're doing that, that you're actually using the correct ratio and you're selecting for long fragments when you're doing your purity. Okay, quantification and quality ratio checks. This one is really important, and like I said, if you don't take anything else home with you, take this home with you because this is really important. You need to quantify your DNA and you need to do quality ratio checks, and you need to hit your targets for these because if you don't, you're going to have problems down the track. It's expensive, it does take a bit of time, and you want to make sure you get this right before you keep going. So quantification, use a fluorometer. Don't use a spectrome because they're not going to be accurate, particularly when it comes to fungi. So for us, we've got a qubit up in Dbook. Use the qubit, quantify your DNA, and aim for a minimum at this point of 100 nanograms per microliter. It's, yeah, you do need a lot. You can get away with less, you can make it work, but if you can get to 100 nanograms per microliter in a 50 microliter solution, you're going to do really well. You're going to be able to get through all the following processes and have enough, because you're going to lose DNA along the way. So if you can aim for that, you're doing really well. Quality checks. So this will be done on the Denovics in Dbook, or you can do it on a nanodrop if you have access to one. 230 ratio needs to be between 2 and 2.2, and your 280 ratio needs to be about 1.8. This is really important. If you can't hit those ratios, you need to do a purification. The other thing that I want to say is that don't use the Denovics in this instance for quantifying your DNA. It's not accurate. It's going to take into account all of the impurities that are in your sample. So if you've got a really low 230 ratio because of all the impurities in your sample, the Denovics is going to include that as DNA and count it towards your quantification. So you're going to find that the Denovics is going to say you've got 300 nanograms per microliter when you've actually only got 100. And this is an example of just that. So this is some work that I did last year with Shona on the minION. It's a couple of samples that we ran through, fungal samples that we ran through. You can see on here that the 280 ratios aren't too bad. They're around 1.5, 1.6. They're a little bit low, so we needed to do something about that. But the 230 ratios are anywhere between 0.4 and 0.7. So you're trying to get that up to 2. So there's a lot of contaminants in there, and this is really typical of what I've seen for fungi. Low 230 ratios and 260, 280 ratios that are pretty close. And so the other thing that you can see is the DNA concentration on the Denovics is saying there's like nearly 600 nanograms of DNA there, which is just not accurate. That's taken into account the 230 ratio. That's what's happening there. So I've found this is really typical for fungi. So moving on to your purification, if you get quality ratios like that, you need to do a high molecular weight purification. This is a kit that I've used just recently this year. It actually worked quite well. I hadn't used it before, but it's from Qiagen. It's a genomic tip kit. Basically just looks like a big pipette tip with a filter in the middle. There's no centrifuging, it's all gravity flow. So they do claim that it's not going to shear up your DNA, so that's what you're looking for. The fragments are sized up to 150 kb with this kit. The average size is between 50 and 100 kb, so quite big. I've used the 20G kit, which is going to be able to purify DNA up to 20 micrograms. So that's a lot of DNA. So if you feel like you've got really great samples and you're going to have more than that, you can buy bigger kits. Just make sure you pick the kit that's actually suited to how much DNA you've got. So if that's all gone well and you've got to that point and you've quantified after your purification and you've still got plenty of DNA there, you can do a short read eliminator. This is really easy. 2 tubes, one is your short read eliminator buffer, the other one is an elution buffer. It's really quick, really easy process. This is really important because, like I said before, it actually removes all of your short reads. So through all these processes, you are invariably going to shear up some of that DNA just because you're pipetting. And so what you want to do is try and remove the short reads. Now this is really important that you check your fragment size. Run a gel, check your fragment size, because you need to know what size your fragments are so that you can choose the appropriate short read eliminator. You don't want to choose a 25 kb short read eliminator if your fragment sizes are only 20 kb. If you do that, you're going to end up with 0, because it's going to get rid of everything. In my experience, I use the 10 kb because my fragment sizes in the past have been about 23 to 25. So I've stuck with the 10 kb and it's worked quite nicely. This is what it does. So you can see in the pink, this is a sample that's been sequenced with no size selection by a short read eliminator. And you can see at the bottom there, the bottom left-hand corner, you can see all of those short reads that have been sequenced. It's resulted in an N50 of 18 kb, which isn't too bad really, but after they've applied the short read eliminator, it's got rid of all the short reads, or a big chunk of them at least, and your N50 has gone up to 41, which is brilliant. So your N50 is telling you, it's an indicator of your short read size, of your fragment size. So that's saying that 50% of your reads are, for the no size selection, 50% of your reads are shorter than 18 kb, and 50% of your reads are longer than 18 kb. So the further up you can push that N50, the better it's going to be for your analyses, and the easier it will be, the more likely you're going to get a complete genome. So this is really important, and you need to consider this in light of how much DNA you've got left as well. If you're getting right down to the bottom end where you don't have much DNA left, you may not actually be able to do this, otherwise you're not going to be able to sequence enough or get enough coverage out of it. So this one, if you've got enough DNA and you're going great guns, make sure you do this one, it'll make your life a bit easier. So this is just an example of a sample that I've put through this process for my PhD. This is Amphomyces. So I started out with an extract, this is PL5, did a high molecular weight extraction, ended up with 118 nanograms per microliter, and it did take me about, I think it was about 4 to 6 months to get this to work, this extraction to work, but we got there in the end. Denovics, put it through the Denovics, got a 280 ratio of 1.7, so that's really good. It's a little bit lower than your 1.8, but that's acceptable. 230 ratio of 0.84, so that's terrible. So had to purify that. There's obviously something in there that is dragging that 230 ratio down, and it's contaminated. As you can see, the Denovics called a quantification of 315, so that compared to 118, that's reading the impurities in the 230 ratio. Ran a fragment gel to have a look at what size fragments I had after my extraction to make sure that they were high molecular weight. And the DNA ladder on that one is a 23 kb, so those fragments are probably somewhere around 23, 25 kb in length. So that's really good. You can see a little bit of smearing underneath that product. They're the short reads, or they're the reads that are getting sheared up. So you can sort of see there we need to do a short read eliminator and try and get rid of as much of that as possible. So if you're not going to use a bioanalyzer, it is expensive, and running this gel seems to be just as effective. A 0.8% agarose, and I've run it at 30 volts, so really low, for about 4 hours. So really low and slow, you really need to be gentle with those long fragments. So I moved on and I did purification, and what happened was it did lose a lot of DNA through that process. It worked really nicely. I ended up with 44 nanograms per microliter after purifying. The 280 ratio didn't really move much, but the 230 ratio is bang on now, 2.2, so that's perfect. And that's what you want to see. And the other really interesting thing is that now the Denovics has actually come down in line in terms of quantifying that amount of DNA. It's in line with the qubit. So that's a really good indication as well that you're coming back towards a really pure sample. What I would say about this kit is that I haven't actually used it too much, but I think there is a lot of room to optimise it so you don't lose that much DNA. So I'll probably have a play with that and try and optimise it a bit more, but I think I think you'll find that you won't lose that much DNA as we go on. It will improve. Once you've purified it, run your gel again. Make sure you haven't sheared up that DNA as a result of doing that purification. So I ran that gel again, and those fragments are still there. It might be a little bit lower, but that looks good. So you want to make sure you still have long reads by this point. So after that, I had enough DNA, so I ran it through the short read eliminator, and it worked really nicely. Lost a bunch of short reads, so you can see that quantification has dropped down to 35.8. Hasn't done too much to the ratios, which is good. And funnily enough, it's even improved the de novo quantification. So it's almost in line now, which is really nice to see. So that gives you an indication of how many short reads you actually had that you've now got rid of, which is great. There's no point in keeping them because you're going to lose out on your sequencing reagents. It's going to take longer. And chances are that when you do your analysis, it's going to bin them anyway, so why sequence them? So 35.8, what I found as a bit of a tip with your short read eliminator, when I first did this kit, I think I only had 25 nanograms per microliter, left it in the fridge overnight, it dissolved a bit further, and the next day I had 35. So it doesn't recommend you do that, but I recommend that if you can, let it sit in the fridge overnight, let it dissolve further, you'll actually probably find you've got more DNA available the next day. So library preparation. So if you can get to this point, in terms of library preps, depending on what application you choose and which library preparation you need to use, there's different amounts of DNA input. So for a rapid kit, for example, you need a total of 400 nanograms, which isn't too bad. For a ligation kit, you need 1,000. So that equates to about 25 nanograms per microliter once you start this. That is an absolute minimum. If you've got less than that, you're really going to struggle to get enough coverage, again, depending on what your application is. So I had 35, so I'm going good guns at this point, so I can start the library prep. So in terms of DNA library prep kits, these are probably the two common ones that we've been running up in Dbook. There's a rapid kit. This one really is designed to use, for example, in the field. It's really quick, it's really easy. They're not technical processes at all, specifically compared to Illumina. These are really easy to get through. With the rapid kit, it's literally a 10-minute library prep and you're loading it onto the sequencer. Illumina can take hours, this takes 10 minutes, so it's really quick. Essentially what happens here is there's a transposome, it's going to cleave your DNA. It's going to attach on an adapter, and then it's going to use that to attach on another sequencing adapter. And that's what the motor protein is going to use to transport it to the nanopore. That takes 10 minutes to do, so it's really quick. The trade-off is that you're not going to get a lot of throughput. So if you need a lot of coverage, if you're doing whole genome assembly, you're not going to want to do this one. The ligation kit is going to solve that for you. It's for maximum throughput. It does take a bit longer. This one, they say, takes 60 minutes. It takes me about 90 minutes to get through. So if you're starting out with this one, I'd probably allow an hour and a half to 2 hours to get through everything and to get towards loading it onto the sequencer. Once you get good at it, you probably get much quicker, and yeah, 60 minutes is probably achievable. This one has an optional fragmentation in it, which sounds a bit odd. If you don't want to fragment up your DNA through all these processes, why is there an optional fragmentation in there? And the thing is that there's a couple of papers coming out now that suggest that if you actually have a really low starting concentration or low sample input, fragmenting your DNA can actually result in more data. Sounds a bit strange, but they're actually seeing now that if you fragment up into smaller pieces, attach your adapters on, sequence them, and then reassemble through your data analysis, you can actually get more through. So that's only going to work in some specific cases, and that's a fairly new thing that they're publishing on now. In most cases, you're not going to want to do that. I haven't been fragmenting it up. So I've been skipping that step. You go straight on to an end prep, DNA repair. You ligate on your sequencing adapters, and 60 minutes later or 90 minutes later, you're ready to load it onto the MySeq. So it's all really, it's quite simple. It's not hard to do the library prep at all. So they're the two kits. Like I said, there's about 56 of them. So the applications are quite wide. If you want to do whole genome sequencing, I'd recommend you use a ligation kit. You need 1,000 nanograms in total. Loading on a minION. There's a couple of steps to loading this. It's very different to Illumina. It's very different to loading a MySeq Flowcell. The first step is to draw out the storage buffer. It comes with the yellow storage buffer. You need to get that out. You need to be really, really careful with these because you can kill them really easily. They're $900 and you don't want to kill them and waste your money. So you do need to be really careful with this. You don't want to introduce air. As soon as you introduce an air bubble through your pipetting, you're going to damage the whole thing. You won't be able to use it. And I've done it. I've done it a couple of times. So it's not hard to do, unfortunately. The next thing you do is pipette through a priming mix. So you're priming the array, ready for the sequencing library. Your last step is to actually load on the library, and that's done in a different port up the top here. So instead of actually pipetting in, like you do in most cases, you actually hover over the top of the sample port and you drop it in. So you've got to aim and drop it in. Through surface tension, it draws it in. It's no problems at all, but you do need to be really careful with that. My recommendation here is that if you've never loaded a minION, don't make your first time when you've done your library prep and you're ready to go with your DNA, because you're going to kill it, guaranteed. I've got some dead flow cells up in Dbook. Come up and practice on them, and I can show you the technique. I can show you how to do it, practice on them, and when you feel a bit more confident, then do your library prep and load your library. You don't get a second shot with these. Once you load your library in, if you kill it, your library's gone. So then you have to start again. So that's my recommendation. Don't make this your first time at loading a Flowcell. So you've loaded your library in, you've started your sequencing run. This is what you're going to get. You're going to start generating your raw data. It's going to start base calling as you go. What I wanted to say about this is that depending on what you're doing, you do get 72 hours of runtime, but like I said, you can stop it. Once you've got enough coverage, you can just stop it. You don't need to keep running and waste your flow cell. So if you're expecting that your run might only take 6 hours before you get enough data to answer your question, be aware that base calling actually takes a lot longer. So you might be expecting a 6-hour run and then you can load another sample on. That's not the case. If you've got a 6-hour run, the base calling component might take another 6 hours. And again, depending on what computer you're running off, it's going to take time to base call. So you might actually need to allow 12 hours for a full run, not 6. And that all depends on how much data, how good your sequencing run is. If you've got a really good sequencing run on, you're going to generate lots of data really quickly, and it's going to struggle to keep up. So we've got a bigger computer up in Dbook, we call it supercomputer. In most cases, it can keep up with base calling. But if you generate a lot of data, it might take another day to finish the base calling. So just allow extra time when you're planning for your actual sequencing run, that this component will take quite a bit longer. Right, so assessing your run. Once you get your run on, it's actually really important that you stick around. I recommend for about 45 minutes and keep an eye on it and see what's happening. It generates all these really cool graphs and plots that you can use to assess your run as you go. So it's all real-time data. And it gives you the opportunity that if you notice that things don't look good, if you've loaded it and you've killed it, you'll see it on this because you won't see any green. It'll be blue. Green is good, blue is bad. So if you've killed your flow cell when you've loaded it, you'll see it here and you can just stop and try and start again. You might actually pick up that something else has gone wrong too, and you might decide, I'm not going to keep on with this and waste my $900 on this flow cell, I'm going to stop it. I'm going to try and start again with my library prep. So this is called the channel panel, and essentially what you want to see is lots of green, like I said. Bright green is really good. It means that those channels are actively sequencing, and that's what you want, as many channels as possible with DNA strands passing through them. The more blue that you see, that means that you've got nothing happening. So there's no sequencing, there's no opportunity to be generating that data. Duty time plot, this is the next one you want to have a look at. This is going to give you an indication of what your channels are doing over time. So this one here has been running for about a day, and this is a really nice sequencing run. So lots of green. You're starting off with about 80% of your pores actually working, which is actually quite a good run. You're never going to get to 100, but 80 is fantastic. And what you see is this really slow and steady increase in inactive channels, so an increase in the blue. That's normal. Over time, over those 72 hours, they're going to start dying and turning off. So you want to see a really slow increase in those inactive channels and lots of green. It's a bit hard to see, but you can probably see between the green and the blue, there's a really faint dark green component. They're the pores that are available but aren't actually sequencing. So you want to make sure that that is a really small percentage of your active channels as well. So this is a good run. Your MUX scan is the next one that you want to check. And what the minION will do is every 1.5 hours, it'll actually do an MUX scan, and it will just completely stop sequencing, and it will check the overall health of its flow cell. So it'll go through, it'll check all of the channels, and it will actively turn some on and turn some off. So in the beginning, what it will do, it'll start out with this MUX scan, and it won't actually turn all of its active channels on. It'll just turn a component on. So maybe 25% of them will turn on, and the others will just remain off. And it does this to kind of protect itself. So if something's going wrong, you're only going to lose 25% or thereabouts. The others are going to be protected. And what it will do as the run continues is it will scan itself every 1.5 hours, have a look at the overall health, and if it needs to turn a few more on to increase sequencing, it will. And it will do that over time. So you can see that through this MUX, there's this slow increase in active channels. And a slow decrease in active channels, and that's inevitable, and that's what you want to see. If you see something just dropping off straight away, you've got something really seriously going wrong. So you want to see a nice linear drop. This is the next one that's really important. This is your read length histogram. This is going to tell you what your long fragments are doing. And this is where we all get our bragging rights from. You want to get the biggest N50 you can to give you long fragments to make your data analysis nice and easy. So on this particular graph, the estimated N50 is 7K. So that's actually quite small, but what it's saying is that the component that it has sequenced, 50% are less than 7KB and 50% are bigger than 7KB. So you want to try and get that N50 up as high as you can. The best I've done is 24.1. So that's the leaderboard. We can try and beat it. But this is an important one, and it gives you an indication of how you're going with read lengths. This is the next one that's important. It's your translocation speed. So this gives you an idea of speed at which that DNA is translocating through the nanopore. You want to hit that green target, and as long as you haven't completely overloaded your flow cell, this should be fine. But like I talked about earlier, if for a number of reasons, if you notice that this drops, you see it just drop and plummet straight off, that means that your flow cell's running out of energy for some reason. What you can do now, like I said, is reload that ATP with an ATP kit. You can load it back in and you'll see it come straight back up into the green zone. So keep an eye on this, and if you notice it dropping off, you'll want to reload it. So what does a high quality sequencing run look like? Lots of green. So this is really nice, lots of active green channels in your channel panel, and a really good duty time plot with plenty of green there. So what you can do when you're assessing your channel panel is you can break it down even further. So as your pores start turning off, when you first start your run, you can actually break it down and you can see why they're turning off. This is really quite clever, and it will tell you why those pores are turning off, and so you can use that to assess why you've got a bad sequencing run. So you might then stop it and go, right, I've got a problem with channel blocking, I need to go back and purify my sample a bit further. So you're not going to waste your whole run. So a couple of problems that you might come across, and I have come across these myself. This is what a low pore occupancy looks like. This is not good. This means that you're not sequencing much at all. There's really nothing there. So you can see on this duty time plot that there's a really small component at the bottom of bright green, and then this big chunk of dark green. And then over time, over a really short period of time, this is over, the drop-off has been 22 hours. All these, the channels are all turning inactive, so they're all turning off for some reason. This is indicating that you don't have enough DNA, so there's nothing going on. You're not generating any data, there's not really much sequencing happening at all. There's probably two reasons for this. You either didn't have enough DNA when you started, or you've lost it through the library prep. So when you've loaded it onto Flowcell, there's not enough DNA there to sequence. The other possibility is that your adapter ligation hasn't worked. So when you ligate on that sequencing adapter, if that step doesn't work and you don't have a sequencing adapter attached to your DNA, you can have all the DNA in the world, it's not going to get sequenced. So you could have a ton of DNA there, but no sequencing adapter ligated on. And that means it's just never going to get sequenced, and it's just going to sit on your Flowcell doing nothing. So you need to have a think about, if you see this, where you've gone wrong, stop, don't let this run for a day, stop it and restart your library prep, try and fix that problem. Another issue that you might see is Flowcell failure due to osmotics or surfactants. So these are just going to destroy the Flowcell. You don't want them in there, you need to have nice pure DNA. And what you're seeing really quickly, this is only over 13 hours, is that the Flowcell is just basically stopped. So instead of 72 hours, you've got 13 hours because those contaminants have got in there and just basically just killing all your channels and your nanopores. So you can see that over a really short period of time, lots of blue. So that's not good. That's something that you want to look out for within the first couple of hours and stop it if you see it. The next one is probably the most common that I've seen, and this is channel blocking. So this is what I was saying earlier, that again, you need to have high quality DNA and you need to purify it. If you don't, this is probably what you're going to see, channel blocking. So you can determine this by, you can see the top dark blue bar. They are pores that are trying to recover themselves. So if you break down why your channels are going inactive, this is what you can see. These ones are becoming inactive because, or they're in a state of recovery on their way to becoming inactive. The reason the reason a pore will try to recover itself is because it's blocked something. And there's lots of contaminants that will get in here and just block those nanopores up, and they won't be available for sequencing. So what these are trying to do is they recognise that they're blocked with something. What it will do is reverse that electrical current. So it's really clever. It'll recognise it. It reverses that electrical current and tries to push it back out. So instead of continually trying to drag something through that's blocking it, it reverses the current, tries to push it back out. If it's successful, it will recognise that, turn the current back the other way, start drawing it back in, and it will go back to an active channel in green and wait for some DNA to turn up. After a period of time, if it cannot recover itself, it'll just turn itself off and become inactive. And that's what you're seeing, or what you will see further down the track, is that dark blue just turning to light blue, becoming inactive. So this is a really short sequencing run. This is less than an hour, and you're just seeing lots of pores, more and more and more, trying to recover themselves. So this is an impure DNA sample, lots of contaminants, and they've just blocked up all this stuff, or they're starting to block up all the pores. If you see this, you want to stop, go back and try and purify your sample further. So this is just an example of myself going through this process, and I actually ran some samples that were not purified just to see what would happen in comparison with the sample that I did purify. And this was something unexpected that I found during the process, that you can see in red at the adapter ligation stage of the library preparation. A purified sample, you can expect to lose about 30% of your DNA at each step. So there's 2 steps in a ligation kit. There's the DNA repair and the end prep. You wash it, and then you do the adapter ligation. So you can expect at both of those steps of the library preparation to lose about 30% of your DNA. And again, this is why you need to start with quite a lot, because you need to expect to lose it along the way. So with the purified samples, I was hitting that. It was about 30% loss when I was quantifying it. So this is another reason why it's really important to quantify your DNA on a qubit through the library prep process. Qubit it after your DNA repair and end prep, and qubit it after your adapter ligation to make sure that you've got your DNA still there. So I saw a 38% loss with a purified product, and with the same product that I didn't purify, I lost 80% of it. So there's something going on there with these samples associated with contaminants and with that adapter ligation step. Not saying that the adapter is not ligating, but something is happening there that means that when you wash it to get rid of any adapters that haven't ligated on, you're actually washing all your DNA away as well. I think that this is a result of phenol, potentially, because that will give you a low 230 ratio. Phenol will inhibit your library preparation steps, which is what you're seeing here. And potentially, if you can purify it and get rid of it, you're actually going to see that you're going to retain that DNA. So this is sort of an example of what you can expect to see if you don't purify and you don't get good quality DNA. I think I did load CES7 non-purified. I loaded it on anyway just to see what would happen, and it was terrible. It really was. So this is an amplemyces sample that I loaded that I had purified, and it went really nicely. It ran for 39 hours. I just let it run to try and get as much data out of it as I possibly could. You can see on the duty time, there's that really nice slow linear drop over that period of time. You can also see that probably the pore occupancy is a bit low. So through this process, I've worked out that what I can actually do is ramp up the amount of DNA that I put in, so I could increase that and get more data out of it. But otherwise, it was a nice run. It started off with 1,549 available pores on the Flowcell, which is the best I've actually ever seen. So this is a really, really good Flowcell, loaded really nicely. And we generated 9.14 gig of data. So that's a fair bit of data for whole genome assembly. And for me, that equated to about 180 times coverage. That was more than enough data really to do the whole genome assembly, so really happy with that. An N50 of 9 and a Q score of 9. So good quality sequencing data. So this was a nice run all in all, and answered the question we needed it to. Here's an example of the same sample that I didn't purify, and it was terrible. It's a bit hard to see on this picture. The PDF report didn't work for this one, so I have to actually take a photo of this on my phone on the screen. It's a bit hard to see, but there's actually a component here. I don't know if you can see. Yeah, you can see that. There's a faint line down through here. Everything under that is light green, and that's what's being sequenced. All that other green there are just pores that are sitting there doing absolutely nothing. So that 80% loss of DNA at the adapter ligation step has caused that, and this really sharp drop off, and that's all the contaminants blocking up all the pores. So it's really quite susceptible to that. So this ran for 68 hours. I just let it go, see what would happen, and we only generated 1 gig of data. So 68 hours, I got 1 gig versus a purified sample that ran for 39 hours and got 9 gig of data. So really, really big difference there. This is 24 times coverage, couldn't really do much with that. You can see on the quality score graph as well, towards the end there it's getting really dodgy, so the quality has dropped right off. It's not a good run at all, and that's what you will see if you don't have pure DNA to start with. A couple of other things that I just wanted to touch on quickly. The Flangle, like I said earlier, we now have the Flangle up in Dbook as well. This is actually really useful. Like I said earlier, you're not going to generate a lot of data out of this, so you may not be able to answer too many questions. This guy is actually really useful in QCing your library. So when I started out, what I actually did was I used this to put my library on, loaded it onto a Flangle Flowcell, started a sequencing run, and checked it. And what I found was that I had a few issues still. And so what I got was some QC data for $90 instead of $900. So I let this run, realised that I had low pore occupancy. So I went back, made some changes, did another library prep, loaded onto the minION. It was a much, much better run. So I got that answer for $90 instead of $900. So it's really useful if you haven't run a sample before on minION or through Nanopore. Start off on the Flangle and QC it. Find out what's not working, go back and fix it, and when you're happy, then run it on the minION, because it's far cheaper to make mistakes on this guy than it is to make it on the minION. All it really is an adapter, so it plugs into the minION and it works exactly the same way. Loading it is a little bit different to minION, to the minION Flowcell, but otherwise it's really easy and I can help you with that. The last thing that I wanted to mention is in-field sequencing. So quite a few people have asked me about doing sequencing in the field. The minION is marketed as a portable device that you can sequence anywhere. Plugs into your laptop, you can take it into the field, it's easy to transport. And yes, that is the case, but I think probably what I've highlighted in this presentation is that it's not that simple. There's still, particularly for us when it comes to fungi, there's still a lot of hurdles you've got to jump over. And again, it depends on why you want to take it into the field. There's no reason that I can think of why you want to do whole genome assembly in the field. I can't think of why you want to do that. It's going to take you weeks to make that work, so there's no reason to do it in the field. There are some other reasons why you might want to go into the field. If you just want to be able to do a species identification in the field for a farmer, that's fine. You might not need much data. You only need to generate a small amount of data really quickly. Sure, that might be the case. But you've got to get over that problem of extracting your DNA in the field. High molecular weight, high purity. So essentially, if you want to make this work in the field, you do need to think about how you can jump over all of those hurdles. And I'm not saying that you can't, because I think that you can, and I think this is a really exciting space to work in and try and get this to work. I think it is possible, again, depending on your application. And I think what you'd want to do is make sure that for that application, you can make it work in the lab before you go anywhere near the field. You might need to forego quality checks, forego some purification or short read eliminators. You're not potentially going to do that in the field. If you want to get a quick answer, generate just a small amount of data, sure. So they're the kinds of things you might need to work on if you want to consider doing this in the field. This is an extract from 2016 where they successfully sequenced some DNA on the International Space Station using minION. And so this was marketed really heavily to show that the minION can be used in the field, but what they don't really talk about is what they did was they just sent the DNA up there. So they didn't send a sample up there, they sent some purified, high quality, really good DNA up to them, and they just did a library prep and loaded it. So that's the difference. And they successfully showed that you can sequence in space and get the same answer when you sequence the same sample, the same DNA on Earth. So that's what they showed, not necessarily that you can jump all those hurdles in the field. And I think we have an extra challenge in that other samples are much easier to sequence. Humans and animals, you can get a much purer sample to begin with. When you're working with fungi, you've got a much harder time. So we've got some extra challenges there. That's all I have to say, and I'm more than happy to take some questions.
Thank you very much for the wonderful presentation. We had a number of questions during the talk from people on Zoom.